Hello everyone, how are you all? I hope so you all are doing well. Myself Sunil Pawar from New Catholic Mission High Secondary School Jhabwa welcomes you back for the new video session of 12th class for biology subject. As you know that we are continuing our 6th chapter, right? The molecular basis of inheritance. As you know that I think so all concepts are also clear. In the previous session we already talked about the genetic material DNA, its structure. We experimentally proved with the help of that Griffith experiment and the others is really DNA the genetic material. We also talk about the structure of the DNA. We also talk about the packaging of the genetic material DNA, right? We also learn about the structure of DNA with the help of the Watson and Crick model of DNA, right? We also learned that what will be the qualities of the genetic material, okay, which helps it to perform the proper working and the functioning. Clear? Then now in this turn, in this second part of this video or this chapter, we are actually talking about the working of the DNA, especially for the replication. How it can replicate? You know that replication means the formation of the replica. Okay, that means the copying of DNA, how it can be performed, and also a simple process of transcription also there. Okay, that is called the central dogma of the protein biosynthesis or the molecular biology. Clear? So just one by one we are learning. We already learned about the DNA and RNA. So first we are beginning with the differences between the DNA and RNA, right? With the previous discussion in which we talked about the structure of the DNA and RNA, clear? I hope so. The concept of structure of DNA and RNA is very clear. They are working and somewhat about their exceptional features too. Okay, so here first in the tabular form we are talking about the differences between the DNA and RNA. As you know that DNA, it usually occurs inside nucleus and some cell organelles. Okay, you know very well that it present where inside the nucleus, but this one some cell organelles also, that means outside the nucleus. Can you recall the cell organelle where this DNA is present? Right, this is chloroplast and mitochondria, right? Okay, so these two cell organelles are there, chloroplast and mitochondria. Mitochondria is present in both animal and plant cell, while chloroplast is present only in plant cell. Clear? Now then this RNA is found only in the cytoplasm. Okay, now then DNA is the genetic material while RNA is not genetic material except in certain plant viruses. As you know that we talk about the retroviruses, okay, and some plant virus, likewise that TMV, tobacco mosaic virus, which is having their genetic material as RNA, not DNA. So few exceptions are there where this RNA is present as the genetic material. Otherwise the functioning of RNA is only to carry the genetic information from nucleus to cytoplasm only. Okay, now then basically DNA is double stranded, but exceptionally it is also found in some viruses like the phase 174. Okay, in this one DNA is single stranded. Remember, this is exceptionally. Otherwise in almost all organisms where DNA is present, it is double stranded. Clear? Now then RNA is the single stranded. Here is also exception, likewise the reovirus where the RNA is double stranded. Okay, remember this single stranded DNA and double stranded RNA is not generally found in the organism as their normal feature. Okay, it is an exception, right? Then DNA is of single type, while if you observe then RNAs are of three types. One, that is the mRNA, second is rRNA, and third is tRNA. mRNA is the messenger RNA, right? tRNA is the transfer RNA, okay, and rRNA is the ribosomal RNA. So there, this way, these three types of RNA is present in the cell, while DNAs are of single type only. Clear? Then The most important difference between the ribose sugar, pentose sugar, DNA is consisting deoxyribose sugar while RNA consisting ribose sugar only. Clear? Then these nitrogenous bases are A, G, A, T, C, G. Okay? This is the adenine, cytosine, guanine and thymine, right? While in RNA, if you observe that, then this T is replaced by U, that is the uracil. Clear? Uracil is actually replacing which one thing? That is the thymine in the RNA molecule. Clear? Then these hydrogen bonds are formed between the complementary nitrogenous bases like A and T, okay, and G and C. You know that A and T is having the double hydrogen bond and the G and C is having the triple hydrogen bond. Clear? While here the base pairing during the hydrogen bond is not actually observed, but in somewhere where it is in the coiled form, then this bonding is there. Okay. Now this DNA is spirally twisted to produce a regular helix. You know that Watson and Crick proved that DNA is a double helical structure. Okay. Now, while on the other hand, in the RNA, if this helix is present, then it is not the true one, it is the pseudo helix. Okay. That means occasionally it found there, otherwise it is absent. Now, DNA can replicate its own molecule. Okay. Exact copies of DNA can be produced, while RNA cannot be formed its copies. Okay. And if RNA want to produce itself, it want to replicate itself, then surely it need to perform the reverse transcription. That means RNA molecule will first form the DNA and then later on DNA will form the RNA, right? Now then this DNA transcribe the genetic information to RNA, while RNA cannot be transcribe information to DNA, but it transfer this information which is taken from the DNA in the form of the translation of the polypeptide chain or a specific series of the amino acid to synthesize the protein or a specific polypeptide chain, right? Then the quantity of DNA is always fixed in a cell, while the quantity of the RNA, all the three types of RNA is varying actually according to the nature of the cell or its metabolic activity. Okay. Now one more thing, always the ratio of the purines and pyrimidines is 1:1 in the DNA, while it may vary because there is no complementary purines and pyrimidines found in the RNA, because there is no complementary strain found against it. Clear? And one more thing, DNA is stable in nature. Okay, DNA molecule is quite stable, it is not changing, so its lifespan and its consistency is quite longer in nature, while RNA is quite manipulating, it's easily get mutant. Okay, and that's why its shape and its nature will not remain fixed for a long period of time, it may be changed. Okay, so this way we can consider the differences between the DNA and RNA, and this comparative study will help you to remember the structure of DNA and RNA also. Okay, now we are moving forward toward discussing this term, that is the central dogma. Okay, it is also called as the molecular guideline of the metabolism. Okay, now simply we can also call it as a doctrine of the protein biosynthesis. Clear? Actually central dogma is nothing special, it is just a specific guideline for the preparation of the polypeptide chain or the protein molecules by receiving the information from the DNA. Okay, so here you can see that this it is said that there is one way or unidirectional flow of information from master copy DNA to working copy RNA, and from working copy RNA to building plant, that is the polypeptide chain. So there are specifically two activities, one that is the transcription and second is the translation. So what is the transcription? Transcription is nothing special, it is just the formation of the mRNA by using DNA strand as a template, okay, and then this RNA will carry this information to cytoplasm, and here by using the cytoplasmic content, it is responsible to translate in the language of polypeptide chain or a protein. Remember these two facts, clear? Then one more speciality of this one thing, that is it is unidirectional. Unidirectional means what? Always this information will flow from DNA to protein synthesis. That means DNA to RNA and RNA to protein synthesis of the polypeptide chain, not in the reverse direction, okay. Now this Francis Crick also proposed the central dogma in molecular biology, which states that the genetic information always flow from nucleus to cytoplasm and DNA to RNA. It cannot be reversed, okay. So remember this one thing, there is the specific path of the flow of the genetic information, okay. What is it? DNA to RNA and RNA to protein, or what you call that is the cytoplasmic content, okay. Now with the help of this picture we can learn this one thing. You can see that DNA is forming RNA and RNA is forming protein. So the formation of RNA from DNA is called as transcription, okay, and this protein sometimes protein is complicated. Complicated protein means what? When the protein is not composed of a single polypeptide chain, more than one polypeptide chains are constituting this protein molecule, then we call it the complex protein, and it is either polypeptide chain or protein, and this process called as translation. Remember, this is transcription, this is translation. Transcription takes place where? In the nucleus, and translation takes place where? In the cytoplasm, remember, okay. So here the second diagrammatic fact can be learned. This DNA replicate itself, DNA to DNA, then it is replication, okay, and then this DNA used to produce the mRNA, this is the transcription, okay, and then this mRNA translated itself in the form of a specific series of the amino acids, that is called as polypeptide chain, or it may be a protein, clear? So this is the speciality of the central dogma of the protein biosynthesis, clear? Now then this one more scientist, that is Tamin, he found that the retroviruses, what are the retroviruses? The viruses having their genetic material as RNA, okay. So this retroviruses perform the central dogma in the reverse form, exceptionally, which involves the reverse transcription by using a specific enzyme that is called the reverse transcriptase. What is the transcription? Formation of RNA from DNA. So what is the reverse transcription? Formation of DNA from RNA, and this this procedure is called as reverse transcription, and for this one thing they are having the special enzyme that is called as reverse transcriptase. So if this RNA want to inflow genetic information, then first it need to form the DNA, and then this DNA, by using itself as a template or as a what do you call that is the mold, it will produce using this RNA, and then this RNA, by using the cytoplasmic content, it will translate itself in the form of this polypeptide chain, okay. So what actually this mRNA is carrying forward from nucleus to cytoplasm? It is nothing special, it is just a specific series of amino information for forming a specific series of amino acid, clear? And this is called as, yes, genetic code. A specific triplets of nitrogenous bases when arranged in a series, then it will be considered like this one, okay. So this transcription of RNA synthesis occurs over DNA, and translation of protein synthesis occurs over ribosomes, okay. So remember, there is a difference between the DNA and ribosomal and RNA there, okay. So what is this feature? We can learn from this one thing, okay. Now moving forward, this is the basic diagrammatic view by which we can learn that how this actually transcription performs. See that this is the DNA, okay, but a protein information is only coded in a specific segment of DNA. What we call it as a gene. What is gene? Gene is a specific segment of DNA which is responsible to coded a specific series of nitrogenous bases for the formation of a polypeptide chain or protein to perform a particular function, right. So, this way this DNA strand is having a particular genetic information in the form of this nitrogenous bases in a specific series, okay. Then by using any one strand of this DNA as a template, okay, it is responsible to coded this information in the form of single strand, that is called as the mRNA, okay. And what you observe, yes, TAG, okay. So, this T is replaced by A is replaced by U, you know that U is the component of mRNA which is absent in the DNA, right. So, this way this specific segment we obtain in the form of the RNA, mRNA, and this process called as transcription. Then this mRNA transferred from nucleus to cytoplasm. Here you can see that this specific group of 3 what we call, that is the triplet, okay, the codon, okay, is responsible to coded this specific amino acid. Likewise methionine, valine, leucine are the serine, okay. So, this particular amino acid is coded by this particular codon triplet, okay, and this way a specific series of amino acid is obtained. You can call it as a polypeptide chain or it may be a protein, okay. So, this is in general the central dogma of the protein biosynthesis also, where first process is which one, that is the transcription, formation of mRNA from DNA, then this mRNA is carrying information to the cytoplasm for the synthesis of protein, okay. So, remember this fact, clear. Now, then here we are going to learn about the replication scheme, okay. DNA, there are actually 3 options for copying DNA to its next molecule, that is the daughter molecule of DNA, okay. So, there is 3 possibility, remember. One is that is the conservative method of replication, second is dispersive method of replication, and third one, that is the semi-conservative method of replication, okay. So, first we are considering all these 3 possibility of making DNA molecule from the parental DNA molecule, okay. So, first we are taking the conservative replication. Conservative means what? There is actually the as it is parental DNA strand will stored in one molecule and the daughter DNA strands in the form of a new DNA molecule will stored in the new DNA molecule, okay. So, you can see that this strand will copy itself in the form of the 2, okay, and then later on you can see that here this parental DNA strand goes in a single DNA molecule, while this copied DNA daughter strand will goes into the next DNA molecule. That means there is no what you call, that is the use of this half strands of the DNA, okay. So, that is why we call it the conservative replication. That means the parental DNA as it is saved and kept in a single molecule of DNA, while the new strand which form by using this parental DNA as a template will stored in the new molecule, okay. Now, here we are considering the dispersive replication. Dispersive means there is no specific place or the attachment areas or the spletion of this DNA molecule, okay. Any part of this parental DNA can be attached or randomly managed into the daughter DNA, okay. So, you can see that here if we observe this diagrammatic presentation of this newly formed daughter DNA, then you can see that there is no any specific pattern of the attachment of the parental DNA is observed, okay. So, we also called it the randomly arranged DNA molecule, okay, and that is why this method is termed as the dispersive replication. Now, the third one, that is called the semi-conservative replication, clear. In this semi means what? Half, okay. That means half part means what? The one strand, okay. One strand of parental DNA is stored in one molecule and second strand will stored in the second daughter molecule, and both these two newly formed daughter DNA molecule will have one parental DNA strand and one newly formed complementary DNA strand, okay. So, this way we can see that this 4 DNA daughter molecule will form in the next stage, where this DNA molecule will have one strand from the parental and this 2 will newly form there only. Okay. So, if we are just shuffling the possibility of this conservation, replication method, then we can easily discard this 2-1. Okay. One, that is the dispersive, and second is the conservative, because there is full of chances of any kind of disturbance in the arrangement of the DNA nitrogenous bases, okay, or the nucleotide. So, that is why there is more possibility of their mutation and to get more variable forms of DNA. But practically we observe that the DNA molecule which we obtain after the replication is exactly copy of the parental DNA strand. Okay. So, there is no changes is observed. So, for this purpose only this semi-conservative replication method is considered. Later we will also prove it with the help of one experiment to clear. So, now here this replication scheme is given here, we can learn from this notes also. So, for replication of DNA termed as semi-conservative DNA, replication was proposed by Watson and Crick. As you know that this model is able to explain each and every activity and behavior of DNA, right. So, first of all the 2 strand would separate and act as a template for the synthesis of the new complementary strand. What happened actually? This DNA strand will loses its helical structure, it twisting, it become parallel in nature, then later on its hydrogen bonds will dissolve and this 2 strand will open up, okay, and each strand will form its complementary DNA strand, and then later on this complementary strand will coil with the opposite and opposite, or the it will pair with the complementary nitrogenous bases with the help of the hydrogen bonds. Okay. Now, so after replication each DNA molecule would have one parental and one newly synthesized strand. Remember this one thing, but we know that always we needed evidences in support of this statement. So, here one experiment was given, okay, which is quite this Masselson, Matthew Masselson and Stale, to prove that this DNA is actually replicate itself with the help of which method? Semi-conservative method. Remember, this experiment is very important from the examination point of view. Okay. So, this 2 question in the first the previous session we learn about DNA is the genetic material. Okay. So, there that Griffith experiment. Okay. And now here this Masselson and Stale experiment to prove that DNA replicates itself with the help of semi-conservative method is very important. Clear? Now, here this experimental proof of the DNA replicates semi-conservatively come first from the E. coli and later from the higher organism. Okay. Likewise the human cells and the plant cells also. Okay. So, this Matthew Masselson and Franklin Stale, they actually proved it experimentally in 1958. So, just with the help of this point we can learn this thing. Okay. So, first of all this E. coli was grown in a medium containing 15 ammonium chloride, that means the radioactively the heavy nitrogen, you know that N14 is the normal one. So, here we are taking the N15. So, this ammonium chloride is radioactively labeled and only the nitrogen source for many generations, and we continued this DNA to replicate it generation by generation. So, whatever the nitrogen atoms which present in this DNA will labeled with the N15 atom. Okay. So, now here we observe all the molecules are attached with this something. So, these heavy DNA molecules could be distinguished from the normal DNA by centrifugation in the cesium chloride density gradient. One who is heavier than this one thing can be separated. Okay. They then transferred the cell into a medium with the normal sodium chloride and took samples at various definite intervals as the cells multiplied and extracted the DNA that remained as double-stranded helix. Okay. Then this DNA samples were separated independently on cesium chloride gradient, and by obtaining the specific density we can consider them. Clear? So, this samples is actually separated at the two interval. First, that is the 20-minute, and second at the 40th minute. Okay, so this double stranded structure is taken, and we observe the hybrid DNA molecule. Okay, so here with the help of this presentation we can learn this one thing. Okay, so what do you observe? This DNA is radioactive DNA because it cultured in the radioactive ammonium chloride solution for generation by generation, and this way all this nitrogenous bases which is present in this DNA molecule will have N15 atoms. Okay, now when we allow to grow in the normal ammonium chloride medium for first 20 minutes, then we will get the exact ratio of 1:1. Okay, because you know that here one strand will have N15 and second strand will have N14. Here is also. Okay, then after 40 minutes when we observe, then is the two molecules of pure 15 and all the four molecule, from all this four molecule, two molecules of pure 14 nitrogen and 2 is the hybrid nitrogen will observe there. So it proved when we obtain it with the help of this chromatographic one, then and also with the help of this separate and the gravitational force, then we observe in the first 28 minutes there is the equal ratio of these two is there, while when after the 40th minute you may find the two strips will observe clearly, which will give you the hybrid nature of this one thing. Okay, so remember this experiment is quite helpful to prove that DNA replicate itself with the help of semi-conservative method. Remember this fact. Okay, so now here we are going to discuss the DNA replication machinery. Okay, in this one mechanism, if you observe, basically what are the tools who is responsible for performing this one thing? That is the biocatalyst. What are the biocatalysts? The enzymes which produce in the cell. Okay, so now here we observe that what are the enzymes which is responsible to catalyze, to initiate, to motivate the process of replication. So if you observe, the main enzyme is DNA dependent DNA polymerase. Okay, because it is used as DNA template to catalyze the polymerization of deoxynucleotides, and the average rate of this polymerization by this enzyme is approximately 2000 base pair per second. Okay, then in a second it can be attached this 2000 base pair in a sequence form. Okay, so this is DNA dependent DNA polymerase enzyme. What is the function? It is responsible to use DNA as a template and catalyze the polymerization. What is the polymerization? Polymerization is nothing special, it is just a process of arranging the monomers in the form of a polymers. Okay, now then this polymerase has catalyzed the reaction with high degree of accuracy, because any mistake during replication would result in the form of mutation. So that's why whenever it catalyze, initiate and further proceeded, then it should be very accurate. Okay, now then this DNA polymerization is an energy demanding process. So deoxyribonucleosides triphosphatase serve the dual purposes. One, that is act as a substrates and provide energy for the polymerization reaction. Okay, now the second one, the many additional enzymes are also required in addition to the DNA dependent DNA polymerase enzyme, that is the helicase, gyrase, replicase, polymerase. Okay, so these all are there. These are actually accessory, these are quite supporting in nature. It is not basically required, but it is helpful in the process of polymerization and replication of DNA. Okay, so here if we observe the most important fact, that is the formation of a replication fork. This is the most important phenomenon which we observe in the DNA replication. Here you can see that this is the replication fork. Okay, in this one you can see that this is actually anti-parallelly arranged two DNA strand. One is 5 to 3rd and second its complementary 3rd to 5. Okay, so at the time of beginning or the initiation, by the motivation of the catalyst, or what you call, that is the enzyme DNA dependent DNA polymerase enzyme, it is responsible to initiate this process from this point. Okay, and here this enzyme is called as the RNA primase. It is called as the precursor. it is called as the rider. Okay, just like that. Then you open up the hook of a chain, then it will split into, okay, you can see that here is this two anti-parallelly arranged DNA strands, one 5 to 3 and second is 3 to 5. Here we can see that it start from this initiating point and here it act as a polymerase enzyme, RNA polymerase, okay, primase also, okay, it is considered a precursor who is responsible to start this procedure, okay. So as it will move forward, it will separate, it will split by breaking or dissolving the hydrogen bond, and these two strands will get separated, okay, it will form. First of all, you can see that it will start from 5 to 3rd end, okay, the formation here, it is in the continuity, okay, while here you may observe that it is from 5 to 3rd end where it form in the form of the small segments, clear. Likewise, it is considered as a Okazaki segments because it was discovered by the scientist Okazaki. So on his name we consider the Okazaki segment. It is not in the continuity, okay. So first this strain will form in a continuation, so that's why this is considered the leading strand, okay, and this is called as the lagging strand. Lagging strand means what? The strand which not equally form, okay, along with this one, okay, and then later on when this Okazaki fragments will form, then later on this DNA ligase enzyme will responsible to connect them, and this way this complementary strand will form, okay. So this rejoining is performed by which enzyme? That is the DNA ligase enzyme, okay. So remember this one thing. Now here we are going to discuss the origin of the replication, okay. Remember two facts. In prokaryotic DNA molecule, it is quite simple and small one. So DNA is always start from one point and continue in a single process till the last end, okay. But what happened? If you observe the DNA molecule of a complex organism, then this DNA molecule is quite longer in nature. It may be from 2 to 3 feet in nature, okay. So if it will start from the one point and continued with the last one, then it will take a long period of time, okay, a long time is we require. So that's why in this eukaryotic one, more than one initiation points are there, okay. So by using this endonucleases enzyme, okay, they are considered the reader one who used to read out this initiation sites, okay, from the middle of this DNA molecule, and they start this from more than one point, okay. So it is called as the multiple initiation sites, okay. So now here this DNA polymerases cannot initiate the process of replication on their own, and also this replication does not initiate randomly at any place in DNA. So there is a definite reason in E. coli and this DNA where the replication of originates, and the reasons is termed as the origin of replication. You can also call it the point of initiation, clear? Now, and due to this requirement, a piece of DNA, if needed to be propagated during the recombinant DNA procedures, that requires a vector, okay, one who is responsible to carry this DNA segment from one to the next one, and the vector provides the origin of replication, okay. So this is the basic feature. Now we are moving to the next point or the topic, that is called as the transcription. And the earlier discussion we learned that what is the transcription? The formation of the RNA by using DNA as a template is called as transcription. What is the difference between the transcription and replication? Replication is the process of forming the replica of the DNA, while transcription is the process of forming a specific segment of DNA in the form of its complementary RNA, okay. And it is just actually the formation of the copy of single strand of DNA just to carry the information to synthesize the specific polypeptide chain or the protein, clear. So now the process of formation of RNA from DNA is refers to as transcription, and only a segment of DNA from only one of the two strands participate in the process of transcription, and the both strands are not copied during the transcription because of this following. Suppose that if this both strand will use to copy and form the RNA, then what will happen? The two RNA will produce, and this two RNA will responsible to produce the different polypeptide chain, and this different polypeptide chain will counteracting and working differently for each other, okay. And also the two RNA molecules so form and it will be complementary to each other, and hence would end up forming a double stranded RNA, leaving the entire process of transcription futile. That means there is a great failure of the process of this one thing. Now what are the transcriptional unit? Okay, so now a transcriptional unit has primarily three reasons. Okay, one, that is the promoter, second is the structural, and third is the coding of strand. So if we observe, okay, so now we consider the promoter. What is this promoter? It is responsible for attaching the RNA polymerase binding site. Okay, RNA polymerase also considered the initiating enzyme who is responsible to start the process of polymerization. Second is the structural gene, is a part of the DNA that is actually transcribed. You know that in the gene there are two type of content, that is the intron and the exon. Okay, so what we now, then the terminator, it marks the end of the transcription. Then the specific point of the this DNA will further break the formation of this RNA. Now the structural gene, so the template strand, the DNA strand with the polarity 3 to 5, then the coding strand, the DNA strand with the polarity 5 to 3, this opposite antiparallel strand, and the sequences of RNA is same with that of the coding strand, and all the references points while defining a transcription it is made with the coding strand. So remember this one thing. First we are talking about the promoters, the region of DNA where the RNA polymerase binds, okay, because for the initiation of the polypeptide chain, then located toward the fifth end of the upstream region of the structural gene. Remember this thing, okay. Then the terminator, you know that what is the terminator? Who is responsible to stop the further which one thing transcription, okay. So the region of DNA that defines the end of the transcription and the located toward the third end of the downstream region of the structural gene, okay, and the site where the termination factor, that is the rho factor, bind to the RNA polymerase, okay. So now here the transcription is in the gene. Now here we are considering what is the gene. You know very well that gene is a specific segment of DNA which is responsible to coded a particular series of amino acid. Clear? Now the DNA sequence is the course of tRNA or rRNA molecule. Then the cistron, remember, cistron is the segment of DNA that contains the genetic code for the single polypeptide chain. This is considered the functional unit of the gene, okay. You know that operon, cistron, muton, and there's all kind, five types there, okay. Now the structural gene could be two types, one that is the monocistronic and second the polycistronic. Very clear from this one thing, where this monocistronic is basically observed in the eukaryotes and the polycistronic is observed in the prokaryotes, where the more than one point can be started. Monocistronic having the two, one that is the exon and second is the intron. What is the exon? Remember, the sequences that code for a particular character and is expressed in a matured and the processed mRNA, okay. Now while the intron, this interrupting the sequences that do not appear in a mature and the processed mRNA, while they are actually coding the RNA, then only this exon segments will copy it, while this intron will just eliminate it from this one, okay. Now then the types of RNA, you know that mRNA, tRNA and rRNA, okay. This mRNA is the messenger RNA who is responsible to carry the information from DNA to cytoplasm, okay. Now, and it is only formed when the information need to carry. Second is the tRNA, that is transfer RNA. It is responsible to attach with the amino acid which is present inactively in the cytoplasm, okay. It is considered as the transporter who is responsible to transfer this amino acid from cytoplasm to mRNA, right. Then this rRNA, ribosomal RNA, This RNA is responsible to bind with the ribosome to form a ribosomal RNA unit to decode the information from the mRNA to synthesize a specific polypeptide chain. Okay. Now, there is the process of transcription. Here we observe that it is basically consisting 3-1 initiation, elongation and termination. Clear? For the initiation, this RNA polymerase bind to the promoter and they start the transcription. Okay. Then the process is catalyzed by the DNA dependent RNA polymerase enzyme and it associates the transiently with the initiation factor, that is the sigma factor, to initiate the process. So, first of all, initiation. Okay. And who is basically responsible, that is the DNA dependent RNA polymerase enzyme. And what is the precursor here? That is the RNA polymerase. Now, second, the elongation. Once it will start, then it need to carry in the form of a specific series. So, this is called the elongation. So, this RNA polymerase use nucleosides triphosphatase as a substrate to polymerize the new strain following the rule of complementarity. That means the specific which one thing, RNA nitrogenous base is just coded against the DNA for the template. Okay. Now then, termination. On reaching the termination region, the RNA polymer is associated with the termination factor, that is the Rho factor. Okay. And as a result of this, the nascent RNA falls off, it gets separated. So, also the RNA polymer is also, and this result in the termination of the process. Here the further coding in the form of the RNA segment will stop. Okay. So, this is initiation, elongation and termination. Okay. Then here this transcription can be discussed in the two different molecules. One is the prokaryotic and second is the eukaryotic. You know that the prokaryotic DNA is quite simple because it is lacking antron, while the eukaryotic DNA is quite complicated where antron and exon both are present. Okay. So, when we are talking about the prokaryotic genetic material, then here you can see that this mRNA does not require any processing to become active because it is already activated. Okay. And even in this case, what is the basic central dogma of protein biosynthesis? First transcription and then second translation, but here it may be this translation can be performed parallelly along with the transcription. Okay. Thus in prokaryote the transcription and translations are coupled, that can be performed together. Here you can see that this sigma factor is responsible to initiate the process of copying this DNA in the form of the RNA. Clear? So, it is responsible to identify and start, that's why it is called the initiation factor, the sigma factor. Okay. Second is the elongation. Here you can see that this replicase and helicase is responsible to unwind and just simplify the twisting of this DNA segment, and then later on this hydrogen bonded will be dissolved, and this way this the sigma factor is responsible to form this RNA by using this DNA segment as a template. Okay. And this way it is further perform its polymerization, that is called the elongation. Finally the termination. Here you can see that this is the Rho factor. Okay. This Rho factor is responsible to identify the codes on the DNA, who is responsible to stop the further coding of this DNA segment, and this RNA gets separated. Okay. Because this RNA polymerase enzyme will stop its working. Now when we are talking about the eukaryotic transcription, then here is this basic stage, the 3 RNA polymerases are present there, RNA polymerase 1, 2 and 3. Okay. This one is responsible for transcribing rRNA. This second is responsible for transcribing heteronucleus RNA segment, and this transcribes the tRNA. Okay. And now when this processing of heteronucleus RNA performed, then this heterogeneous nucleus RNA, it contains both the introns and exon. Clear? Then this RNA splicing is there, who is responsible to separate them. These introns are removed by this process and exons are jointed together. And finally the capping. What is the capping? A methyl guanosine triphosphate residue is added to the 5th end of the heteronuclear RNA. And finally the tailing. After 200 to 300 adenyl residues, these are added to the 3rd end of the heteronucleus RNA. and finally the fully processed heteronuclease RNA, now called the mRNA. So what is the difference between the mRNA and DNA segment? mRNA is only the strand which is formed by the copying of exon part of the DNA segment. Okay, it is just lacking the antron one. Remember this one thing. Okay, so now here with the help of this diagram we can learn this thing easily. Clear? See that this is the eukaryotic DNA. Here you can see that. Okay, this is the promoter site. Clear? Here you can find the alternately arranged exon, intron, exon, intron, exon, intron site. Clear? When the transcription takes place, that is responsible to remove this intron one by one, and finally this splicing is responsible to form a continuous segment of mRNA which is consisting only and only exon. It is lacking intron. Okay, so that's why you can see that this mRNA is quite smaller or shorter than the DNA segment. Clear? And then later on this mRNA, by using the tRNA and rRNA, is responsible to form this amino acid chain which is further coiled in the form of the protein. So this is the process of which thing, the transcription. Okay, so in this video we are just restricting our description. Also in the next we will talk about the next procedure, that is the human genome project and the DNA fingerprinting and also this one thing. So I hope that you learn all this fact. Just go through this video and this content twice and thrice. Just go through the proper which one thing, that is the PDF which is provided. Okay, so just learn this one. So till that, thank you very much.